everyone, welcome to Biochemistry class. In this video, I want to discuss one of the biomolecules that we need for our body to generate energy. They are carbohydrates. In the first part of carbohydrates, I want to discuss about carbohydrates function and monosaccharides, their structure and stereoisomer. Okay, let's find out. When you hear the word of carbohydrates, what do you have in mind? A yummy fried rice, spicy noodle, or a delicious toast? Well, you're not wrong. In fact, there are many functions of carbohydrates. They are main sources of energy. There are also plasma membrane components. They can limit our calorie intake, maintaining our heart health. They determine the body's glycemic index and helping keep our digestive system function properly. Before I discuss more, I need to tell you one thing, that sugar, saccharides, and carbohydrates are different names for the same molecule. Carbohydrates molecules consist of carbon, hydrogen, and oxygen atoms. The molecular formula for glucose is C6H12O11, while sucrose is C12H22 and O11. Therefore, it appears that the ratio of hydrogen to oxygen atoms is 2:1. As a result, these molecules were named carbohydrates, derived from carbon, anhydride, or water. In fact, carbohydrates do not contain water molecules, but the name carbohydrates are still used, in addition to other names saccharides. So, carbohydrates can be defined as polyhydroxyaldehydes or polyhydroxyketones, as well as compounds that produce them in the hydrolysis process. There are three classifications of carbohydrates: mono, di, and polysaccharides. Let's learn about monosaccharides first. Monosaccharides are the monomeric units that polymerize to form polysaccharides. The simplest carbohydrates are aldehydes or ketones that have two or more hydroxyl groups. The empirical formula of monosaccharides is CH2O, and N is equals to more than 3. The simplest monosaccharides have 3 carbon atoms, namely trioses, glyceraldehydes, and dihydroxyacetone. Glyceraldehyde is an aldose because it contains an aldehyde group, while dihydroxyacetone is a ketose because it contains a ketone group. We can name the sugars according to the number of carbon atoms that they have, which will be monosaccharides with 4 carbon atoms are called tetrose, 5 carbon atoms are pentose, 6 carbon atoms are hexose, and 7 carbon atoms are heptose, and etc. Combining with the functional groups that they have, which are aldehyde or ketone, the sugar can be named D-glucose is an aldohexose, D-ribose and 2-deoxy-D-ribose are aldopentoses, and D-fructose is a ketohexose. Each of the carbon atoms in sugar that have both hydrogen and hydroxyl group will have a chiral center or asymmetric center. And the convention for drawing linear monosaccharides is to use Fischer projection. The D and L notation are made because of the presence of carbon atoms with configuration asymmetric as in glyceraldehyde. If on this carbon atom the hydroxyl points right, it's D sugar. If it points left, it's L sugar. When we look at this, it is important to understand that with Fischer projection, horizontal lines are implied to be wedge bonds, while the vertical lines are implied to be dash bonds. But how if the sugar have more than one asymmetric carbon atoms? The notation of D and L are specified the asymmetric carbon atom farthest from the aldehyde or ketone groups. So we will call the figure in the left-hand side is D-glucose and in the right-hand side is L-glucose. In nature, sugars are mostly found in the form of D-isomer. Sugar in the shape of D is a mirror image of sugar in L shape. Both sugars have the same name, for example, D-glucose and L-glucose. D-glucose and L-glucose are enantiomer pair. The presence of carbon asymmetric in sugars causing stereoisomer. The number of stereoisomer is 2 power n, where n is the number of asymmetric carbon centers. So, aldoses with 6 carbon or hexose have 4 asymmetric centers, therefore have 16 stereoisomers. There are 8 D-shaped sugars and 8 L-shaped sugar. These are 16 stereoisomers of glucose. These molecules have all the same molecular formula. They are C6H12 and O11, but they differ in the arrangement of carbon and hydroxyl groups. These are examples of 8 D sugars and their common names. So their name will be D-allose, D-altrose, D-glucose, D-mannose, D-gulose, D-idose, D-galactose, and D-talose. The terminology of D and L sugars is a bit more outdated than the R and S that we use to assign absolute configuration. But the convention of D and L sugars has stuck around and it remains that way. However, you need to learn monosaccharides EUPAC nomenclature. If the sugars are aldehyde, the nomenclature based on the aldehyde nomenclature. For examples, for the glucose, their IUPAC name is 2R, 3S, 4R, 5R, 2, 3, 4, 5, 6 pentahydroxyhexanol. D-glucose has 4 carbon chiral center in C numbers 2, 3, 4, and 5. So 2R, 3S, 4R, and 5R, and has 5 hydroxyl groups in C number 2, 3, 4, 5, and 6, and has 6 carbon atoms. While L-glucose is a mirror image of D-glucose. Therefore, its name is 2S, 3R, 4S, 5S, 2, 3, 4, 5, 6 pentahydroxyhexanol. Assigning R and S configuration to Fischer projection is assigning priorities to the 4 substituents according to the Chan-Ingold-Prelog rules. You should remember these rules from organic chemistry lecture. What about D-fructose? D-fructose is a ketose and has ketone group. Its nomenclature based on the nomenclature on the ketone nomenclature. So, the name of D-fructose in EUPAC nomenclature is DS4R, 5R, 1, 3, 4, 5, 6 pentahydroxyhexane 2-none. D-fructose has 3 carbon chiral center in C numbers 3, 4, and 5. So, their name is 3S, 4R, and 5R, and has 5 hydroxyl groups in C number 1, 3, 4, 5, and 6, and has 6 carbon atoms. For L-fructose, which is a mirror image of D-fructose, you should know their name. Okay? Monosaccharides exist in equilibrium between a linear and acyclic form, with the cyclic form being highly preferred. Monosaccharides can form a cyclic form through the formation of hemiacetal and hemiketal formation. This mechanism we learn in organic chemistry. The formation of hemiacetal and hemiketal formation based on the fact that aldehydes and ketone can react with alcohols to form hemiacetals and form hemiketals. These are the formation of hemiacetal and hemiketal. Alcohol attacks the aldehyde and ketone groups and form hemiacetal or hemiketal. In the case of sugars, the hemiacetal and hemiketal formation are intramolecular because the hydroxyl group and the carbonyl are in the same compound. These are the hemiacetal formation or cyclization in the D-glucose as an examples. Electrons from hydroxyl group in C number 5 attack the carbonyl group in C number 1, causing the linear form become cyclic. This can do from either side. Therefore, it's generating 2 different stereoisomers, or we call it as anomers, alpha and beta. Beta anomer when CH2, OH, and OH are on the same side, or when OH groups points up. The sugar is now called beta D-glucopyranose, since it has 6-member ring. The ring structure here is called Howard projection. These are the hemiketal formation or cyclization in the D-fructose. Electron from hydroxyl group in C number 5 attack the carbonyl group in C number 2, causing the linear form become cyclic. This can also do from either side, and it also generating two different stereoisomers, or we call it as anomers alpha and beta. Alpha anomers, it's when CH2OH and OH are on not the same side, or when OH group points down. The sugar is now called alpha D-fructofuranose, since it is 5-membered ring. Okay, now we are repeating again the process of ring formation in aldose as in the D-glucose as an examples. The ring structure of D-glucose causing two different anomers, alpha and beta. Alpha when hydroxyl or OH points down, and beta when OH points up. And this is we repeating again the process of ring formation in ketose as in the D-fructose as an examples. The ring structure of D-fructose causing two different anomers, alpha and beta. Alpha when OH points down, and beta when OH points up. When we draw a cyclic structure of monosaccharides, we sometimes use Harvard projection, but due to tetrahedral nature of the carbon bonds, pyranose sugar form a chair or a boat configuration depending on the sugar. The depiction of the chair configuration of glucopyranose above is more precise than the Harvard projection. This to remind you the chair and the boat form of ring structure. A stands for axial and E stands for equatorial. We should also note that monosaccharides don't always remain in the alpha or beta form. There is an equilibrium between them since the hemiacetal formation is completely reversible. The alpha glucose might go back to their linear form and then cyclize again to form beta glucose. In fact, for glucose, beta anomer is preferred. This phenomena is called as mutarotation. Mutarotation is a change in the optical rotation of solution due to a change in the equilibrium between alpha and beta anomers. The alpha and beta anomers are in equilibrium and interconvert through the open form. The percentage of alpha anomer is about 36%, beta is 64%, and in the very small amount is in the linear form less than 0.02%. So, we know now about monosaccharides, the linear form, and the way they cyclize, and it's the cyclic form that is able to polymerize to form long polysaccharide chains. Let's learn about di and polysaccharides in the next video.